And it appears those firefighters have nothing on Mrs. Kettle, who single-handedly climbed the tree to retrieve her own kitten. Good for you, Mrs. Kettle. Our next segment is one of our in-depth field reports, where one of our most prestigious reporters, Mark Scott, has an in-depth inside experience with metabolism. Let's see what Mark has in store for us.
In day-to-day life, a person may do many things that they rarely think about: breathing, swallowing, and sometimes even eating. What we eat determines how we function and how much energy we have. Carbohydrates are one of the forms of energy that we eat daily and comes in the form of glucose, fructose, sucrose, and many other mono and disaccharides. But how much energy can it provide us? What is our max? And is one saccharide better for you than another? These questions and many more have been asked by an avid researcher in the field named Sarah Sucrose. I sat down with Sarah to find out just what carbohydrates were all about.
Sarah, tell me a bit about your work.
Well, I work primarily with carbohydrates, how they function as a source of energy either exogenously or endogenously to provide energy to the body in the form of ATP. Carbohydrates are oxidized within the body by glycolysis and are completely oxidized by the TCA cycle and the electron transport chain.
Interesting. And where do these processes occur?
Well, it happens in every cell of your body, carried out in the cytoplasm and the mitochondria.
Now, what carbohydrates are you specifically interested in?
Specifically glucose and fructose, but also sucrose, which is a disaccharide of these two monosaccharides.
What exactly happens to these two molecules when they are oxidized?
Well, glucose can be phosphorylated in the cytoplasm by hexokinase in order to convert it to glucose 6-phosphate, which then continues through glycolysis, whereas fructose has two different points of entry. In the liver, it is first phosphorylated by fructokinase to fructose 1-phosphate, or F1P. F1P is then cleaved by F1P aldolase into dihydroxyacetone and D-glyceraldehyde, which are two substrates that can enter glycolysis.
Oh, okay.
In the kidneys, it is converted to fructose 6-phosphate, which is then directly inserted into the glycolytic pathway.
Well, if fructose provides our body with more options, why don't we just primarily ingest fructose?
Well, the oxidation rates of fructose are lower than that of glucose, mainly because it has to be converted into an acceptable form before it can enter into glycolysis. At the end of the cycle, both glucose and fructose can yield 38 units of ATP.
So now where does your research enter into all of this?
Well, my research has to do with the oxidation rates of these carbohydrates. There was a conclusion made by another researcher in the field that there was an upper limit to the rate of glucose oxidation of 1 gram per minute, approximately.
And you thought this rate could be exceeded?
Yes, most of these tests studied only the use of single monosaccharides, whereas my colleagues and I proposed that through a combination of saccharides, a higher rate could be obtained.
I had a chance to catch up with one of the athletes who was part of all three of the studies, Butch Boxington.
I was one of the 8 trained male cyclists. I'm 30 years old and I weigh 75 kilograms. Most of the other athletes were around my age and within a kilogram or two.
Now, what tests did they run on you?
They measured my VO2 max, my individual maximum power, and my volition exhaustion through incremental studies. The actual testing required 120 minutes of cycling at 50% of my max power, while ingesting whichever of the carbohydrate concoctions they had assigned to us. The solution with fructose and glucose had it in a ratio of 1:2 in order to give us the maximum intake possible.
Now, how did they ensure that you weren't running on glucose that was already present in your body and that these trials weren't different from other trials?
Well, they had us eat the same meals before each test, and all of us ran a glycogen depletion training session a week before the testing.
Now, was there any discomfort experienced from any of these concoctions, tests, or anything like that?
Well, there were definite gastrointestinal pains felt after the testing. The higher the glucose level, the more pain that I experienced. The fructose and glucose combo had the best effect. The highest glucose solution made me feel exceptionally bloated, causing cramping and nausea.
How did they measure your blood glucose levels?
They had to do blood extractions for that.
How do you think this information will affect your performance and eating habits, personally?
This will definitely change what I eat before competitions and practices to help me keep my energy up. This should also affect athletes worldwide at any level of competition. It never hurts to have more energy when competing.
Now, after doing all your tests, what were the results you found?
Well, there were 3 tests run in the span of 3 years. The first was in 2003, and it tested the exogenous oxidation rates of glucose by itself, and then with either sucrose or maltose. Sucrose is a disaccharide of glucose and fructose, whereas maltose is a disaccharide of glucose.
Interesting.
We found that for glucose and the glucose and maltose, the same maximum rate of oxidation was found, with it was 1.06 grams per minute. But with glucose and sucrose, the rate was measured at 1.25 grams per minute.
Which is already 25% higher than the previously established upper limit of 1 gram per minute. What were the following studies testing?
The second test in 2004 measured the oxidation rates of glucose at medium and high levels of intake and glucose and fructose ingested simultaneously. We found that the oxidation rates of glucose at medium to be 0.8 grams per minute. At high, it was 0.83 grams per minute, and the combination of glucose and fructose together was 1.26 grams per minute, which was higher than our previously established values.
So again confirming that by combining carbohydrates, you could reach a higher level of oxidation.
Yes, exactly. The final test was another study of glucose and sucrose, testing how the oxidation rates changed with medium and high levels of intake of glucose in combination with sucrose. The results showed a 0.77 oxidation rate for glucose alone, 0.98 for sucrose, 0.9 for medium glucose and sucrose, and 0.2 for high sucrose and glucose.
Now you may ask yourself, why would the combination of different sugars change the rate of oxidation so significantly? We had one of our studio doctors, Heidi McHealth, an expert on metabolism, break it down for us and answer just that question.
The rate of carbohydrate oxidation depends on the rate at which the carbohydrates can be absorbed in the body. The different carbohydrate oxidation rates when combining fructose and glucose is due to the fact that glucose and fructose are taken up by different transporters.
So there are separate transporters for glucose, fructose, and sucrose?
For glucose and fructose, yes, but there is likely no disaccharide transporter. Sucrose and maltose are hydrolyzed to their respective monosaccharides at the brush border membrane. Glucose is taken up by SGLT1, which is a sodium-dependent transporter located at the small intestine. The sodium gradient is what helps propel the transport of glucose through the transporter.
And these transporters are always present in the small intestine?
Fructose is absorbed through facilitated diffusion down a concentration gradient, but glucose is unable to transport through this transporter.
So how does this increase oxidation?
Actually, the expression of both GLUT5 and SGLT1 is upregulated by the presence of carbohydrates in the lumen of the small intestine. When ingesting the maximum amount of glucose, the SGLT1 transporter becomes saturated and cannot transfer at a higher rate. By using GLUT5 to transfer fructose, there's another transporter that can reach saturation, meaning that overall more energy will be produced.
And are the saturations of these transporters measurable?
Well, they can be measured indirectly through measuring lactate levels within the body. Higher levels of glucose in the body means more anaerobic respiration, which means the production of more lactate from lactate dehydrogenase. Lactate levels have been significantly higher in experiments involving the intake of fructose and glucose. These graphs show the results for the 3 studies of glucose and lactate levels in plasma. As you can see, glucose levels have increased across during the first 15 minutes. This indicates absorption across enterocytes by the transporters in the intestine and occurrence of anaerobic respiration. After 15 minutes and up to 120 minutes, the levels of both metabolites are approximately stable as transporters are saturated and carbohydrate oxidation continues.
Now, what are the future directions you could take with this research?
Well, from here we could investigate what effect the amount of salt ingested has on the uptake of glucose. We could also try and look for a non-nutritive supplement that would increase the expression of SGLT1 and GLUT5 in the body in order to maximize sugar uptake.
So if you are looking to optimize your athletic performance during high-intensity training, you can increase your levels of energy production comfortably by ingesting a combination of glucose and fructose in high levels. Because at elite levels of performance, every little bit counts. Back to you, Max.
Thanks for that in-depth report on metabolism, Mark. I hope all you young athletes at home who are paying attention are now going to eat a more balanced meal of carbohydrates every day. In recent news, Europe has been taken over by a flock of cats.
Line up super early just to eat. Monday hot dog, Tuesday taco, Wednesday hamburgers and chocolate milk. Thursday sloppy joes and burritos in the bag. Friday was pizza day, the best day of the week. It always came with salad and a side of cold beef.
Oh, that was amazing.
All right,
Oh my god, that was so